In this brief presentation, we'll take a journey through multiple sclerosis, neuroimmunology, and beyond. And as the title suggests, we'll be focusing on B cells of the immune system and their contribution to multiple sclerosis. We will emphasize the importance of different compartments of the disease, as well as the involvement of B cells throughout these different stages, and the biologies that are involved, both underlying relapses and non-relapses and progressive disease. My disclosures are as follows. In multiple sclerosis, as seen in this MRI axial flare sequence, the disease is identifiable with multifocal lesions that evolve over time, that coalesce, and are associated with tissue loss. This is therefore a dynamic, multifocal, and diffuse process with potential for destruction of tissue. And while the textbooks still teach us that this is a white matter demyelinating disease, we now know that there's a very important involvement of gray matter as well, and that the biologies contributing to the central nervous system injury involve both inflammation and mechanisms of degeneration. In this cartoon at the top, we're looking at the typical clinical course with an onset of relapsing remitting MS followed by what has been called secondary progressive MS. At the bottom, on the same timeline, we're looking at two brain imaging parameters. The area under the curve in green is the total volume of those T2 hyperintense lesions that I had shown you on the previous scan, and they can be considered as a measure of the burden of disease as it evolves over time. You'll notice that earlier in the course, this burden tends to increase, but later in the course, there is less further increase in this measure of disease burden, yet if you look at the top, patients continue to get worse. So there is an important disconnect. The other disconnect is represented by the little black arrows at the bottom of the x-axis. Imagine that each represents a time point where you would expect to see a gadolinium-enhancing lesion, which we think of as local breakdown of the blood-brain barrier integrity and active focal perivascular inflammation. There are more episodes of active perivascular inflammation earlier in the disease, fewer and fewer later, yet patients again continue to get worse. To be able to explain these dissociations between the clinical and the imaging courses in MS, we have to accept that there must be at least two different biologies contributing to injury. The blue biology and the red biology, which historically had been referred to as inflammatory versus degenerative, but probably the updated way of thinking about them is that the blue involves important aspects of peripherally mediated immune cell activation, insufficient regulation, and trafficking into the CNS around vessels, so the perivascular inflammation underlying relapses. In contrast, the red biology, which we do not understand as well, seems to involve processes that are compartmentalized within the central nervous system. And these CNS compartmentalized processes include both inflammation and tissue degeneration as the biological substrates of progressive non-relapsing multiple sclerosis. In this cartoon, we have a simplified model of the MS immunology, the immune pathogenesis. In the lower right, you have the target organ, the central nervous system, separated from the periphery by different barriers, such as the blood-brain barrier, or BBB. And the concept is that activation of immune cells in the periphery, upregulation of molecules in these cells that enable them to more efficiently traffic and get across the barrier, and then get reactivated in the central nervous system, where they participate in the injury process. And while simplified, this is nonetheless a useful model to think of the different sites in the cascade where current and future therapies may work. Historically, MS has been considered to be a T-cell mediated disease. This is a focus that was in many ways propagated based on animal modeling of MS, the most commonly used animal model being experimental autoimmune encephalomyelitis, or EAE, in which you can inject an animal in its periphery with a piece of the brain, such as a piece of myelin. The animal will have activated T cells from the periphery that attack the central nervous system. And if you took from a sick animal only those T cells that recognize the CNS and gave them to a healthy animal, the animal comes down with the same disease. So in the animal model, this is a powerful way of demonstrating that all you need to get this disease is CD4 positive T cells that recognize the central nervous system. Probably EAE is a better model of acute disseminated encephalomyelitis, particularly in a post-vaccination context, and there are important differences between EAE and multiple sclerosis. Nonetheless, EAE has shaped the thinking of MS as a T-cell mediated disease, and when we think of the B-cell contribution, for the most part, the B-cells are thought to contribute through antibodies, just as they continue to teach us in the medical textbooks. The early attempts to target B-cells through anti-CD20 were based on the idea that maybe there are bad antibodies made by the B-cells, and indeed we know that such antibodies exist in the central nervous system of patients, and whether removing the B-cells as precursors to these antibody-producing cells would translate into benefit. It's important to note that from an immunology perspective of regulating autoimmune disease, the presence of B cells and T cells that can recognize self is actually true for everybody. It is normal. This is called normal autoimmunity. This includes central nervous system directed T cells and B cells that can recognize many targets in our CNS. Usually, they are kept in check through the balance that is established between effector and regulatory arms of the immune system. On the T cell side, we recognize multiple effector subsets like Th1, Th17 type cells, and regulatory T cells, or Tregs, that are able to downregulate effector cell responses. And it is the important balance between them that regulates normal autoimmunity. We also know historically that T cells do not respond in isolation. They need to have their antigen presented to them by antigen-presenting cells, such as myeloid cells. These include dendritic cells and macrophage, possibly microglia, and they too can come in different subtypes and flavors. And we've known over the years that there are important interactions, two-way interactions between T cell subsets and myeloid cell subsets, such that different T cells can shape the responses of the myeloid cells, and different myeloid cells can shape the responses of T cells. This therefore deserves to have a bidirectional arrow between the T cell subsets and the myeloid cell subsets. As far as B cells are concerned, what might they do in this context of regulating autoimmunity? For the most part, historically, B cells have been considered relatively passive cells that wait until they get help from T cells to turn into antibody-producing cells. And if they're involved in protection, it's because they make protective antibodies. If they're involved in disease, it's because they make autoreactive antibodies that can bind to self, targets, and cause injury. And indeed, in multiple sclerosis, we have known for many years that there are abnormal antibodies in the central nervous system of MS patients, the increased synthesis rates of immunoglobulins, the presence of oligoclonal bands as examples. It is with a view of trying to target B cells as producers of antibodies that the field started looking at targeting B cells with anti-CD20 therapy. This shows you the lineage of the developmental steps in B cell development, showing you from the early stem cells in the lower left all the way around and back to the plasma cells. CD20 is expressed on a broad range of these cells, but not on the stem cells or very early B cells, and not on the plasmablasts and antibody-producing plasma cells. We're showing here the very impressive impact of removing B cells, in this case in the two OPERA studies, OPERA 1 and OPERA 2, comparing ocrelizumab to the 3 times weekly subcutaneous interferon beta. And what you can see on the left, results of OPERA 1, on the right of OPERA 2, the y-axis depicting the development of new brain lesions by MRI, and the different epochs or time periods in the study on the x-axis. You'll appreciate that compared to the treatment with the interferon beta, where some new lesions were observed, there were very, very few lesions, a very substantial decrease in new lesion development with ocrelizumab relative to the active comparator. This was a magnitude and an effect that surprised many people in a field where MS has been considered to be a T-cell mediated disease. When we and others looked at the spinal fluid of patients treated with anti-CD20 antibody, we found that the abnormal antibodies present were not impacted at a time that patients were benefiting from a very substantial decrease in new disease activity. This meant two things. One is that B cells are clearly important in the development of new MS relapses, but that this role of B cells did not depend on antibody production. And this is part of what the field recognizes as important antibody-independent roles of B cells, both in health and in diseases like multiple sclerosis. In this paper, we suggested that abnormal cytokine responses that we and others could show are expressed by MS patient B cells may contribute to the abnormal pro-inflammatory responses of the T cells. And indeed, we could show that removing of the B cells by anti-CD20 from patients with MS resulted in decreased inflammatory responses of both CD4 and CD8 T cells. So the T cells care about whether B cells are present or not. And when we go back to our traditional view, we now recognize that B cells can come in multiple different types, producing different profiles of pro or anti-inflammatory cytokines. They can regulate autoimmunity, and they can talk back to T cells. So this also deserves a bidirectional arrow, now indicating that B cells can influence T cells. And finally, in additional work, we were able to show that B cells can also shape the responses of myeloid cells, such that we can complete this triangle with interactions between all these different subsets, defining the immune response state as relevant to multiple sclerosis disease activity in patients at different points in their course. This discussion has focused to this point on the blue biology and how peripheral roles of B cells are implicated in affecting the inflammatory cascades that lead to new relapses. B cells, overly activated, overly pro-inflammatory in MS patients, contributing to pro-inflammatory responses of both myeloid cells and T cells. What about the red biology, the CNS compartmentalized biology? Here we know that one of the important pathologies relates to the subpial cortical injury, where there is demyelination, neuronal loss, and microglial activation, which occurs in a region underneath the CSF and not in a distribution that is perivascular. It requires us to think of a different mechanism of injury. And this subpial cortical injury is rather important because it actually correlates better with progressive disease outcomes, both cognitive and physical, than the white matter lesions do over time. Also in pathology, we appreciate that the meninges of patients with MS may have immune cell infiltration, in some cases rich in B cells, and that there has been an association of the presence of such immune cell collections in the meninges and this type of subpial cortical injury, again shown here in red in the middle figure. Also importantly, when one looks at the pattern of pathology of this subpial cortical injury, and shown here in the right-hand graph, are on the y-axis neuronal loss, so percent reduction of neurons. Big numbers means more neuronal loss. And on the x-axis, we're looking at the different layers of the cortex, from superficial layer 1 on the left side all the way to deep layer 6 of the cortex on the right. And you'll appreciate that there is a very substantial reduction of neurons, particularly in the superficial layers of the cortex, and there is a lesser degree of abnormality as you go deeper and deeper into the cortex. The same pattern of abnormal response is seen for microglial activation, where the greatest microglial activation is in the superficial cortical layer 1, adjacent to the CSF, and then diminishes as you go deeper into the cortex. This raises the intriguing hypothesis that perhaps factors secreted from immune cells within the meninges, perhaps from B cells as well, may contribute by diffusing to the cortical interface to the subpial cortical injury in these patients, and therefore perhaps play an important role in non-relapsing progressive disease biology, or the CNS compartmentalized process. Some observations which support this view include recent observations that soluble products of B cells of MS patients can be toxic to both oligodendrocytes, as shown at the top, and to neurons, as shown at the bottom, as compared to soluble products from B cells coming from patients or healthy controls who do not have multiple sclerosis. Altogether, the suggestion is that immune cells within the central nervous system, including perhaps CNS compartmentalized B cells, may be supported there in ways that are important for us to understand and may contribute to the underlying subpial cortical pathology as a biological substrate of progressive disease. This does not mean that degenerative processes are irrelevant. It is more likely that there's a combination of these processes contributing to the disease process. So altogether, we've emphasized the different compartments in multiple sclerosis, the blue versus the red biologies, namely the relapse biology and the progressive non-relapsing biology, and have suggested that B cells of the immune system may contribute importantly in these different compartments and to the different processes of multiple sclerosis.